The very last step now of our operating pipeline for fNIRS would be sharing the results, visualizing and communicating what we've done. We all know how to talk about our data, write out which stats we use, do Bayesian whatever or frequentist reporting in APA format. But there's also this importance of visualizing our data. The picture is worth 1,000 words mantra. Visualizations communicate the point you're trying to make more quickly and intuitively if done well than text. And color and shapes are also more engaging to look at than a lot of text. It'll attract more people to considering your paper, your poster at the conference than just a wall of text will. So think carefully about what message you're trying to send about your results, and then you can make a cool and engaging visualization for it. Visualizing point estimates will be the most simple thing that you can visualize. Point estimates are single values, often meant to be characteristic of a wider set of values, such as an average across time, an average across subjects. And at its simplest, visualizing this needs to show the value of that point estimate pretty easily. But a good visualization shows much more info than just that. It would be a comparison with another group, dispersion of underlying values that point estimate is summarizing, or the number of underlying values. So for instance, on the right there you can see a bar graph that only shows the values of 5 different measurements. It doesn't show you any sort of confidence interval, doesn't show you how many data points are making up that estimate, doesn't even show you what those different bars are supposed to mean. Whereas at the bottom, that's what's called a rain cloud plot. It shows you lots of different ways in a pretty format what the axes are. I cut off the actual axes labels, but they're there. It'll show you the distribution of the two sets of data you're trying to consider, how many data points are making that up, etc. You'd want to include any text that's necessary on a visualization, but you want to also try to keep it as brief as possible. You want to make the picture be able to speak for itself. With fNIRS data, you would be able to use this approach to communicate, say, beta estimates from a GLM model, synchrony estimates for an entire time course, etc. There are many, many tools out there and programs available for communicating this, since this is what all scientists have to do at some level. Some popular options would be, for simple plots, you could use SPSS, Excel, MATLAB, or R. Some more sophisticated options would be specific kinds of plots called like rain cloud plots, violin plots, forest plots, etc. Some of the best programs for plotting things, in my opinion, are ggplot in R or matplotlib in Python. And then I also use some MATLAB packages, but they're not as well supported, I think, as the other stuff in R and Python. If you're visualizing changes in values, not just one value, you'd use this if you care not about the value of an estimate, but how it evolves over time. The more complex the results are, the more a good plot can communicate it better than text. So if you're doing anything more than a point estimate, it can be difficult to just say what is going on in like a text of your results section, and can be much more fruitful to make a really informative plot, because our eyes can detect shapes in data traces better than we can understand how like a mathematical function is shaped if we just give the formula. You can use plots like this to show time courses of values, and maybe a separate plot to show mathematical values of those changes. So for instance, on the right we have 2 brain time courses, and below that is a moving window correlation estimate between those 2 time courses. And this would include dispersion displays on different time points if it's not too visually cluttering. This visualization that I provided here has a lot going on visually already, so it's not going to provide dispersion points. But if you have something that has, say, fewer time points, or the field's not already so cluttered, you can kind of give maybe confidence intervals around each point. You could give a sort of partially transparent colored haze around the line to indicate what that confidence field would be. With fNIRS data, you would want to use this approach to communicate what a neural time course looks like, what moving window estimates would give you, etc. And then you can be visualizing a lot of data. So you would be using this if you have many, many variables to express, or many variables to express over time. Also a case where individual values themselves aren't as important, but you care about values relative to each other, or values clustering in certain ways. You want to utilize color as well as shape for good visualization of these sorts of plots. For 2D data, you can use things like waterfall plots to show the point estimates of many different variables that you were estimating. You can use heat maps like the bottom, which we already talked about for doing state dynamics. For 3D data, you can do gradient maps like on the bottom right. In fNIRS data, you would use this approach to communicate things like state change dynamics, or the point estimates of lots of ROIs. And then there's visualizing network data. So networks require special visualization considerations. You'd want to display things either as a hairball, like above right, or kind of heat map of communities, how certain nodes in the network are clustering. You can show them visually like below. You want to think about, when you are trying to visualize a network, about 3 things. First, what kind of structure of the entire network do you want to display? Are you trying to order it in a certain way? What shape do you want it to have? Do you want it to be really stretched out? Do you want it to be kind of more clustered together? You want to think about the depictions of the edges too in your network. Is the size, color, thickness, or distance going to communicate the strength of a connection? Are you going to use different like dashing versus solid line conventions to communicate maybe type of connections? And then think about the depictions of the nodes in your network. Again, size, color, and shape can be used to communicate maybe centrality of a node, its community membership, etc. Some popular network plotting tools would be SOCNetV, which is a standalone executable, Gephi, GGNet2 in R, iGraph in R, NetworkX in Python, GraphTool in Python. I personally don't do a lot of social network analysis, so I don't use these tools a ton, and I don't know all of them or what the absolute best ones are to use. So you can talk to the Parkinson Lab people who do much more of this for their data. A note on color I want to make: when you are visualizing your data, just remember that around 10% of your audience is going to be colorblind. So choose colorblind friendly contrast colors. If possible, you also want to use colors that have culturally learned meaning. For instance, bright colors tend to evoke perceptions of large numbers, strength of quantity or value. So if you're doing something like a heat map, use like bright yellows, for instance, for the top of your data range, and cooler colors like blue for the bottom of your data range. And if displaying data from different groups or different variables on the same plot, use different colors, of course. You want to disambiguate things as much as possible. The place I took all of these recommendations from, and that has many, many more recommendations, is this data viz cheat sheet I found on Twitter that's really informative. So if you have these slides, maybe really zoom in onto this to read through it. It'll give information on like why you want to be communicating things, planning how you would visualize data, some basic principles of effective design, the effectiveness of various things, selecting the right base graph, etc. And if you're just watching this video, maybe pause it and zoom in on some things to look at it, because there is also a part 2, so you want to stop and look at this too. I find this graph really, or this cheat sheet really, really useful. I always refer back to it whenever I'm making new plots for papers and such. Besides just visualizing your basic data though, you might be wanting to visualize brain data specifically and depicting point estimates on a visual brain model so that you can communicate the spatial locations of those results. This requires having data for all spatial locations that you can plot, as well as coordinates of those locations. If you don't have the MMI coordinates of your data, and you only have their relative locations compared to each other, you can make topographical 2D heat maps like this, where each number is a different channel from an fNIRS measurement, and this particular heat map is showing synchrony values in channels with like sort of a linear interpolation smoothing function applied to it, so that it has nice kind of pretty heat maps to look at. If you have the 1020 coordinates of your channels but nothing else, you can still project channel values onto an estimate of where those would be located on the brain surface. You could use this just to communicate data on what looks like a brain if you want to be able to show a brain picture in your publication, but don't use this to determine if your data falls in a very particular area or not. This wouldn't be accurate enough for that, since you're kind of guessing what is the most likely internal location of your external point. Also, you really shouldn't be doing this with fMRI either, because of the issues with like other people's brain morphology not being the exact same as each other, or different conventions of warping data to a common space in a processing pipeline. So really in fNIRS and MRI, you shouldn't be trying to say, look, this picture is on this gyri, therefore it is absolutely this gyri. But with fNIRS, like, definitely don't do that because of this extra qualification I mentioned. Though if you do have the MMI coordinates of your data, say you did a 3D digitizer, or you have a good 1020 to MMI conversion atlas, or you did a full fMRI structural image where you could register external locations to someone's particular internal locations, then you can more accurately plot this data onto the brain surface. Though again, remember the qualifications about fNIRS, I should say spatial specificity, positioning on external landmarks, etc. These can be blocky presentations like the screen you saw before, where that's communicating just individual channel estimates, or you can use linear interpolation to smooth between channel estimates. The tool I'm using personally right now to communicate activation on the brain surface for my fNIRS data would be a combination of using XJView, and that's in MATLAB, and this standalone program called Surfice. So first, if you're going to use this process, you want to acquire a full version of XJView. That's an MRI data visualizer written in MATLAB, and it includes an fNIRS specific package. So go to that link there to download XJView. And then also download Surfice, a standalone surface renderer. I like this one because unlike most MRI surface renderers, it doesn't unfold all of the sulci and gyri. With fMRI, that makes a lot of sense to do, because then you can just look at a more flat representation of the brain and see how activation spreads through that. But with fNIRS data, our signal is not always penetrating to the deepest parts of sulci, so if you unfold the brain all the way, representing that data on the brain surface will be very patchy, and like data will be missing from what was in the sulci, and will be only little spots that were over the gyri. And so you want to leave the brain folded up. And then the Surfice just makes it look nice and shiny and pretty and colorful. And so it also runs on both a PC and a Mac, which is nice, since I am using a Mac right now. And there's another URL for downloading that. Once you have those two softwares downloaded, you need to have your MMI coordinates ready of each of your channels. Again, either through an MRI structural, a 3D digitizer, or the 1020 to MMI conversion atlas. And represent this in MATLAB as an n/3 matrix, where n in rows is each channel you had, and the 3 columns would be each dimension of the MMI coordinates, the x, y, and z. Also prepare your values to visualize as an n/1 column vector in MATLAB. These can be whatever you want to visualize. They can be t-scores, they can be beta values, correlation values, etc. Then in XJView, use the NIRS to image function. This will require a few arguments. The image file name argument will be whatever you want your file to be called when it gets generated and put out by this function. MNI is the MNI matrix that you just prepared, the n by 3 matrix. Value is your n by 1 value column vector. DoInterp is a Boolean argument where one is saying I want there to be linear smoothing between the colors of each channel or no. Do XJView is just, do I want to open this up in XJView first? XJView is some rudimentary surface rendering as well. I usually just say no because I know exactly where I want to visualize it over in Surfice. And then bilateral is another Boolean that asks, is this MMI Coordinates bilateral? Were you looking at the same arrangement of channels on the left and the right sides of the head? If yes, this would just be doing no interpolation between what is the left and right side of the head. And then once you hit that function, you generate both an HDR and an image file. These are possible to open in any MRI surface renderer that takes these file formats. But since we're using Surfice, what you would do there is open the program, select overlays from the top menu, and then add overlay. You want to make sure you're closing any overlays in between different images, or else you'll add multiple overlays and they'll kind of overlap each other and look weird. You also want to be opening, when you're adding an overlay, the particular image version of the file and not the header, because it doesn't know how to read the header file. Then you have an overlay like the top right pop-up. You want to use the menu on the left to change any attributes about this map that you'd like. You can change the heat map colors. You can threshold your image to only consider certain values that are above a certain threshold. You can change the brain color. I believe the default brain color in Surfice is actually like a pink brain, and so you might want to change it to gray to make the values of your activation stand out more, and so you can do that also in the menu on the left. And then there's a little square or like a cube at the bottom left of the image that if you just grab that with your mouse and move it around, it enables you to rotate the brain across all 3 rotation dimensions, and so you can rotate it to looking at the particular view of the brain that you want. And finally, once you have the picture that you like, you would use File, Save Bitmap to save your images as a .png file. Another tool I've used in the past for visualizing brain data, but I don't use so much anymore for reasons I'll say right now, is NewsLab. I don't use this anymore, except for very particular circumstances, because it's kind of a lower quality rendering of a brain image. It's only got blocky channel presentation too. But it has the ability to create videos of your activation over time. And so I might still use that if I want to be able to represent, say, 2 brains activating in a way that's visually identifiable, and then a video is kind of really convincing to show that in like a presentation. Another downside of this is that it requires MATLAB 2014a or earlier. And it runs as an executable on PC, but only as encrypted MATLAB files on Mac. So you can't edit or look at the code that it's using under the hood. For using NewsLab to visualize data, you can do a basic preprocessing and general linear remodeling in NewsLab itself. If you want to do your own stats of like creating beta estimates or doing any sort of analyses like synchrony, you can do that on your data on yourself and then create what would be like a dummy analysis of some data file in NewsLab. So you'd be going through all the steps of preprocessing and specifying a first general linear model, but it would be nonsense. It wouldn't be anything that you're actually trying to care about. But it sets up the data format that we would then go and edit. So that would be the first step for representing data in NewsLab. Then once you create this data and specify a first level model for each subject, it will make an SPM map. You would want to open the fNIRS SPM struct in your MATLAB window. You only need to do this for one subject since you're trying to represent one brain. Open the fNIRS SPM struct, then NIRS, then beta, and then that will open a matrix that contains your dummy beta estimates for every beta you specified in the first level model, which will be on the rows, and every channel columns. Make sure that when you are trying to create a dummy brain like this, that there are no channels marked as bad in the dummy specification. That would result a 0 in the beta's matrix that you then can't change. So for instance, this matrix here has a lot of zeros, and then I would have to use a different, or go respecify the brain so that none of the channels are being removed, so that those would be numbers that I can actually change. And then finally, if you have that matrix ready, you just go in and retype those beta values to whatever values that you want to be displaying. They can, again, be t-scores, your real betas that you calculated, correlations, etc. Make sure that file saves, and then back in NewsLab, open SPM level 1 and then view. Select the NIRS SPM file you edited, choose which beta line you want to display, the one that you made changes to, and then click view. In a window it pops up, you can get a depiction of your activation displayed over the brain. You can turn on from here 3D rotation so that you can again get the particular view on the brain that you want. You can rescale the activation colors. You can choose the map style. The default when this window opens is going to be actually a two-dimensional like EEG map. And so you would potentially want to change that to either like a head surface rendering or an MMI brain surface. And it allows you to save the image as a JPEG. If you want to do any thresholding of your data here, you'll have to make sub-thresholded channels have 0 values when you're editing the betas, because there's no ability to threshold here. You can also save brain activity as videos, and you don't need to go through the whole SPM level 1 specification for this part. And that's, I think, the part that breaks in newer versions of MATLAB, so this may work for you to make brain activity videos if you have a newer version of MATLAB. To do this, you would go in, you would just have to load in your raw data in NewsLab, create some sort of dummy event file if you don't already have one. You can just put like a stimuli mark at the very beginning of the study that lasts like 5 seconds and then save that. You don't have to consider that stimuli mark ever again. And then from there, you can go into the data viewer boxes at the top right of the NewsLab home. You would load in the NIRS info file you just generated, and then click Map Hemodynamic States. The window here is, again, you can turn on 3D rotation, rescale the color bar, choose map style, and view images of time points or of the animation. For instance, this is a video that I made using this method with NewsLab. This would be when I was trying to communicate kind of neural synchrony while two people are watching the same really engrossing video. This is a fighter pilot run where at one point things get really, really exciting. Woo! And the brains do the same thing. And so with a video like that, it's very easy to see that synchrony happening in the brains, whereas if you're just saying that in text, it might not be as convincing and engrossing. There are some other tools for visualizing fNIRS data that I have not used for publication yet, so I don't know them very well. I think I've attempted almost all of these, but for one reason or another I found them really difficult or kind of limiting. NIRS SPM is one that does surface rendering, but the sort of fidelity of the brain is not super high, and it looks like just like a flat topo mat had been laid over the brain instead of rendering it onto all the gyri. NIRS Analyzer has a visualization, but I just found that difficult to use. I think I have to learn more about the way it does the estimation of where on the brain surface external landmark would be, because that's the part that I think I was most confused about. NIFRI, or however you pronounce it, is another one where you can do your own analyses and then just put those values onto a brain surface rendering. I haven't used this one, so I don't know how easy it is to use. And then there's Atlas Viewer, which is a companion to Homer 2. I have not used this one because it requires certain toolboxes in MATLAB that are additions beyond the standard base MATLAB, so I just don't have those toolboxes, and so I couldn't run this. But with that, we're now done with the bootcamp. Thanks so much, everyone, for watching and listening to this and spending all your time at this bootcamp. Again, I'll go through the preprocessing code walkthrough in a separate demo. This is the walkthrough of the analysis scripts I've already written, so feel free to visit my GitHub for downloading that code and demo data, and check out the other lectures that are on those specific walkthroughs. Besides that, see you later, I guess. Thanks!